Welcome to our new video, exploring the world of gene therapy. We'll discuss gene therapy and its applications in healthcare. Gene therapy involves introducing, altering, or replacing specific genes within a patient's cells to correct genetic diseases. In this video, we'll delve deeper into gene replacement therapies. Gene therapy falls into two broad categories based on whether it targets germ cells or not: somatic gene therapy and germline gene therapy. Somatic gene therapy specifically targets non-germ cells, ensuring that introduced genetic changes do not pass down to future generations. Germline gene therapy modifies genes in reproductive cells or embryos, potentially leading to heritable changes and raising ethical and safety concerns. Most clinical trials currently concentrate on somatic gene therapy. Now let's look at the differences between in vivo and ex vivo gene therapies. In vivo gene therapy, the genetic material such as therapeutic genes or gene editing tools is administered directly into the patient's bloodstream, muscle, or other specific tissues using vectors like viral vectors or nanoparticles. In vivo gene therapy is commonly used in conditions like muscular dystrophy, SMA, hemophilia, or specific eye disorders where the target cells or tissues can be effectively modified within the patient's body. In contrast, ex vivo gene therapy involves the extraction of cells or tissues from the patient, followed by genetic modification in a laboratory and subsequently reintroduction of the modified cells back into the patient's body. Ex vivo gene therapy approaches are commonly applied to hematopoietic stem cells and are particularly effective in the treatment of blood and immunological disorders, including certain types of leukemia and lymphomas. Gene therapies are commonly delivered using viral vectors. A commonly used viral vector is adeno-associated viruses, AAVs. AAVs have advantages like low pathogenicity, long-lasting gene expression, low risk of mutagenesis, and versatility. The main disadvantages of AAVs are their low cargo capacity and the potential for immune responses. Lentiviruses are used in ex vivo gene therapy. Lentiviruses offer advantages like larger cargo capacity and integration into the host genome. They carry a risk of inserting new genetic mutations and have the potential to cause immune responses. The choice between AAVs and lentiviruses depends on the specific disorder, target cells, and therapeutic gene size. Now, let's look at some of the common side effects of gene therapy and how it can be managed. Immune-related side effects are the most significant side effects of AAV, adeno-associated virus gene therapy. This can occur when the patient's immune system reacts to the AAV vector or the therapeutic transgene. This may present as liver toxicity with elevated liver enzymes, transient drop in platelet count, or less commonly a condition called thrombotic microangiopathy leading to renal impairment. Immunomodulatory treatments like steroids and close monitoring of patients help to minimize such side effects. Please note that patients with pre-existing immunity to a viral vector are excluded from receiving gene therapy using that virus. Approved gene therapies are available for various neurological conditions, including spinal muscular atrophy, SMA, cerebral adrenoleukodystrophy, CALD, aromatic L-amino acid decarboxylase, AADC deficiency, and Duchenne muscular dystrophy, DMD. However, gene therapy isn't limited to neurological disorders. Conditions like hemophilia, immunodeficiency disorders, and numerous other medical conditions also have gene therapies available now. Gene therapy holds great promise in revolutionizing healthcare. As research advances, the future offers exciting possibilities for improving patients' lives. Thank you for joining us on this journey through the world of gene therapy for healthcare professionals. Please do not forget to like and share this video. Also subscribe to our channel for more informative videos in the future.